Hello, my name is Helena. I'm going to further explain the engineering logic applied to biological systems. So we could say that one of the main characteristics of our species is our great capacity and will to transform the environment. We have been studying and characterizing basically the components of the natural world in order to first understand them and then manipulate them to use them for their own benefit. When you look at our history, we could say that studying inert inorganic materials, such as this rock, was relatively easy. The rock has specific properties and behaviors that we can predict depending on its composition, on its shape, its temperature, and other parameters. What we call engineering is the application of this very precise and exact knowledge that we have acquired to invent, build, improve tools and machines that have a specific human function. Our capacity to use and transform materials such as the rock is basically what we call engineering, and we have reached a level of sophistication of manipulation of these materials that has allowed us to build the infrastructure, the tools, and the machines that define our contemporary life and our modern technocultural environment. The computer that we have here is a perfect example of the power of engineering. When we think of all the processes that it runs in parallel, the number and the size of its components, the networks with which it can interact, when we think about all this, is quite overwhelming. However, engineers are realizing that despite all our technology and our capabilities, biological organisms have fundamental properties, features, and components that exceed by far anything man-made. Let's have a look at this plant and look it in the eyes of an engineer and consider it as a machine. This beautiful machine has apparatuses called leaves that are optimized to perform photosynthesis, which converts basically CO2, thanks to energy light, into organic matter, something that this computer cannot do. The plant can also reproduce. It can replicate using flowers, which are basically a reproductive apparatus, quite complex. By ripping this flower, I just triggered a self-maintenance mechanism that will repair the plant. The plant will not die. It can self-repair. It has tools such as roots that can sense every single component of the soil in order to extract what it needs at a specific time. At the micro scale, it has millions and millions of cells. Each cell has a specific function, and it's coordinated, and everything is coordinated with each other. When you look inside the cell, you find molecular apparatuses that are quite sophisticated. This is the ATPase synthase, which is basically converts chemical energy into mechanical energy and then back to chemical energy in the form of ATP that will be used by the cell to perform all kinds of tasks and processes. The engineering logic just enables us to extract the plant from its natural context and to decompose it into individual components that have unique characteristics. This means that in theory, we could manipulate each of these components in order to develop technologies for human benefit. In other words, we can apply the engineering logic to biology. However, biology is complicated. We just talked about it. Biology is messy to the eyes of the engineer. Living systems don't behave predictably like the rock, and the inner workings are difficult to grasp. We don't know how this plant works, especially. In contrast to the mineral world, our understanding of the underlying mechanisms of biological machines is relatively new. We just discovered DNA in the 50s, for example. For a while, our interventions and capabilities were slow and limited. Nevertheless, we had a lot of fun with the biological playground. Just by experimenting with it, we were able to adapt crops to our agricultural needs, for example, or even aesthetic needs. Like this colorful strain was developed by a farmer in the US just by cross-feeding some species. We have used our empirical knowledge of nature to create environments compatible with our industrial life. Intensive breeding and farming of both animal and vegetal life forms on a planetary scale are key components of the Anthropocene and of our contemporary natural landscape. Recently, considerable technological efforts have been employed to apply the engineering logic to biological systems even further. Over the last couple of decades, we have dramatically expanded our technological knowledge and capabilities, and we have increased the speed and the scale of human control over these biological machines. Why? Today, the field of biological engineering considers that at the heart of biology, there is a digital code in the form of DNA, A, T, Cs, and Gs. This means that everything that is happening in the plant is encoded by this DNA, in theory. It means that on the one hand, we can identify a process inside the plant and find it into this DNA when we read it. And it also means that if we can rewrite the code, we can program the plant. Thanks to our ability to both read and write DNA, the field of synthetic biology considers that biology is fundamentally designable. Life becomes a design substrate. As reading and writing DNA is becoming incredibly cheap, the industry is considering life as the most powerful manufacturing technology of the future. I would just show you how my research in theory can expand pharmaceutical manufacturing. So in nature, you can find all kinds of compounds that could be potentially used to develop a drug. Imagine that we all discover that this plant produces a compound that can save millions of lives. What we would do in the past is to try to grow massively this plant in order to extract the drug. Today, in the genetic engineering age, what we will do is to modify this plant to make it produce and optimize this production process. But what we will do now, most likely, is that we will try to read the DNA of the plant and find the specific program inside it that will encode the production of the drug that we're interested in. Then what we will do is either we extract the DNA of the plant, or we will simply buy it online. You can today buy for very cheap DNA that will come in this tube. This is a tube of DNA that I use in my lab. In the synthetic biology age, we consider DNA as and any single process that happens in the plant as a programmable feature that can be transferred into other organisms. So instead of growing the plant, we will take this DNA and we'll introduce it into microorganisms that are easier and cheaper to grow, such as E. coli that I just put here. By introducing the DNA of the plant into E. coli, it will acquire the ability to produce the drug, and then we will design the bacteria to produce a lot of this antibiotic. We will grow them in flasks like this and monitor how much drug is being produced until we find a super producer that is compatible with our needs. Once we find the super producer, we will put it into big bioreactors, which are the bigger version of what my colleague just showed you. And with this, we will finally be able to bring the drug into the market. This example illustrates the bio-utopian manufacturing future that characterizes the vision of my field. A future where the application of the engineering principles to life allows the advent of an environmentally and sustainable industry. An industry that goes in hand with our wet dream of a green revolution. This logic is currently expanding to different sectors of the industrial landscape. It's not just the pharmaceutical industry. We're using it to produce more food, to produce more energy, to produce more materials. In a few years, you might not even notice how synthetic biology is shaping your life. You might wear the same clothes, except that the material and the dyes will be produced by microorganisms. You might eat the same burger, except that the meat will be produced in vitro, and the cheese on top will be produced by microbes as well. We're now entering an age where we're able to imagine and design life and to adapt it to our needs with more control. One could perceive the advent of synthetic biology as a real shift in the way humans perceive and interact with the natural world. In reality, nothing has changed. Our interactions and motivations remain the same. Our connection to these life forms is purely exploitative. We still seek to benefit an already established industrial setting based on profit and on the expansion of human control. Synthetic biology is the result of our impressive technological progress, which is driven by our goal to optimize and to reduce the economic and environmental costs of the technosphere. Our motivations and objectives in terms of exploitation of the natural resources haven't changed. What has changed is the scale of our needs. Before we could produce something with crops, now we need microorganisms to produce it, because it's no longer viable to use crops. The technosphere is looking for potential solutions to sustain our modern world by shifting our focus from the microscale, the scale of the plant, to the molecular level, the scale of the gene. I will now end my talk with some open questions. As the engineering logic will continue to expand to the living world, it is our responsibility to critically think about its implications at all levels of society. How do we want to define our relationship and interactions with this new synthetic kingdom? Do we only want them to use them as factories? Can we use them for other purposes? Now that we're modifying life, what are the boundaries between the natural and the synthetic? What belongs to the natural world and what belongs to a corporation, for example? If we push the engineering logic to the extreme, can we think of ourselves as machines? I believe it's the responsibility of bioengineers like me to expose the inner workings of the technology and to explain you its transformative power. But I also believe it's the responsibility of everyone, including everyone in this room, to engage in the debate that will define the way we will transform the living world. Thank you.
Well, thank you, Helena. That was fascinating and intriguing, and I have more questions than I'll have time to ask you. So I wanted to start by talking a little bit about some of the analogies you use throughout your talk. And one of the things that I talked about yesterday on the Switches panel is the long history of the ways in which we've thought about the relationship between life and machines, right? And you could start in 1948, but you could also go way back to classical thinking, or stop at Kant, depending on where you want to draw the line. The point is, I noticed you use language like program, like machine. You ended by asking, can we think of ourselves as kinds of machines? You talked about encoding DNA. And I'm curious, given my fascination with this language, whether there's a way out. Is there a way of practicing synthetic biology, being a bioengineer, without thinking about life as like a machine? Or did the two go together?
Well, that's a hard question. Well, I think the analogy of machines in the contemporary context is because we found a way to describe some living systems mathematically. Like we try to write equations that would allow us to tell how a system would behave or not, and in some cases it works. It works in the way that it can let us understand how certain things interact with each other. Like mathematics help us understand the world, not in how it's going to happen, but just find certain relationships, like is this protein going to interact with the other one, or things like that. And in that case, it works. And so everyone goes excited, and everyone said, oh, then if we can describe with mathematics, then it means that we can control, because you can change the parameters, and then you can change the equilibrium, and then you can push the system into a specific end. I think that was like what excited the engineers. I think now as a bioengineer, what we need to embrace is the context, the concept of context, and the idea that your organism is very dependent on a lot of things, and that it might change. And yeah, we need to stop obsessed with control, I think.
Yeah, so I mean, what I hear you saying is sort of, we don't need to think about life as a machine, and life might not necessarily be machinic, but that thinking has helped us go a long way in synthetic biology. But perhaps now we're reaching a moment in which there are other analogies that might take over and press design in new directions.
Exactly. It helps us understand of thinking as a machine, because we can simplify a system. We can make abstraction of many things because the plant is very complex, right? So if you think of like, okay, I'm just going to try to write a mathematical bonding between these components, that rationale can help us understand the machine, but we shouldn't consider it as a machine in per se.
Right.
Indeed.
Right.
So, yeah, I think the field has to move into a new direction, and I think it's something very fascinating and complex because we don't know what's going to happen.
So that actually leads me to another question I have, which is about this long history of synthetic biology, right? Which we've heard multiple times this afternoon. The idea that synthetic biology doesn't start in the last 10 years or 50 years, but actually this is something that goes back and is continuous with the Neolithic Revolution, that there's a difference of degree but not of kind between the development of agriculture or animal husbandry and what people like you do now with plants, with microbes, etc. And I hesitate to bring this up because I don't want to make a facile comparison that will make people think that I'm making this analogy. So I'm just going to say up front before I even say it, I'm not comparing synthetic biology to Monsanto. But up until a few years ago, if you went on the Monsanto website and you looked at, they had a timeline for the company, and if you clicked on it, you would think that it would start with the foundation of the chemical company Monsanto in the 20th century. It did not. The timeline started with the origins of agriculture 12,000 years ago. And so there are obvious political and ethical and social reasons for making a claim about continuity, right? So in some ways, yes, obviously, we could say that biotechnology and bioengineering is the human manipulation of the natural world, and we've been doing that for as long as humans have been around. On the other hand, there is a way in which we could also talk about novelty. That there is something that is new, that is different, that needs to be at least thought about or responded to or managed or policed differently than growing,
exactly. There needs to be a certain, at least a certain sense of governance in what we're doing. And the thing is, like, all these technologies are happening so fast, and everything is like going so fast. Like when I started my career, like what we're able to do today, I'm talking about 5 years. It's really crazy. Like the DNA, the amount of DNA that we can synthesize it's insane. And so we're all like kind of like running against the clock, like, wow, what we can do is amazing. Let's do more, let's do more, let's do more. And we're not really actually thinking about, okay, is what I'm doing going to have is my technology actually going to actually really help the world? Like we were saying, the technology of Monsanto is not a bad technology. It's the use of the technology that they're doing that it's kind of like, that is not the most...
I mean, as an anthropologist and historian of science, I have a hard time separating the physical technology itself from its uses. But that being aside,
I understand.
I guess my broader question is, what are the possible social, political, ethical ramifications of making claims to either novelty on one hand or continuity on the other? Is that something important?
Well, I think it really depends on which technology you're talking about, because for some things there is novelty, like when I was like in vitro meat, right, or this kind of stuff. That is really new, for which we have no reference. Making new strains of crops, and you kind of like have to put it back into its context because it involves farmers, it involves a lot of kind of like other actors for certain things. I think it's really like context dependent, and that's why it's so difficult for the public to see that not everything is black or white, because there,
absolutely,
it's a much more complex problem, and I think it's great that people like you are trying to retrace that history so that it makes people understand where does it come from and what is really new, what is actually not, what can be potentially harmful, what is actually just an analogy or just a concept that is used to get more money or more hype. So
speaking of hype, I think you coined a term a few minutes ago, the bio-utopian.
Yeah.
And I wanted to ask you more about that, right? So I should say that in advance of this event, I was asked to respond to a question, what is the world going to be like in 2048? And I drafted an email response that said, the Earth will be destroyed by 2047. No one is going to exist. And then I didn't send that email, but now I've announced it to all of you. So forget about my pessimistic thinking. I want to return to your bio-utopianism. If the best possibilities of synthetic biology come to fruition, what are the affordances, the possibilities, but also what are the limits? If we imagine 2048, what does a synthetic future look like for you?
Yeah, like again, I think we first need to solve our own social political problems. Like the technology can bring many, many benefits. Like we can produce and discover drugs, for example, that can potentially keep us safe from all kinds of diseases. We can also find cure for cancer, to be super cliche, etc. Like we can improve food production, etc. But who is going to have access to that technology? That's something, right? Is there going to exist inequality in that? I think it's like with everything. Like if I want to be very optimistic, I think we will all be bioengineers at some point, because all these biotechnologies will be, you can do many things in your own kitchen. And so I would love that one day people engineer their own life, and they live like they become conscious that they're part of a very beautiful natural world. And like, for example, the microbes that live in your body, you can actually learn from them how they can change your body, how can they improve your health. And so I would love to see this bio-utopian future as everybody embracing the fact that we can actually interact with the living world, and that we can actually benefit from each other. That would be great. And if everyone can have like access to this information and use it for their own benefit, and not that it's only something that is in the labs and in the corporations and only the very specialized people, that would be actually pretty awesome.
So I hear you saying two things. One is that there won't be any technological fixes for social problems, right? So that utopianism has its limits. But also that perhaps there will be a democratization of the kind of work that you now do.
And so that people find local solutions. I think we're never going to find a global solution. Technology is never going to find global solutions. I think technology can bring solutions at the local level. What a community might need in Mexico is very different from what a community might need here, etc. So I think, if technology can bring that, and biotechnology can bring that, that would be pretty cool.
So Helena, from Googling you, I found out that not only are you a very successful scientist, but you are also the developer of a game called Hero Coli. So I want to hear all about what Hero Coli is. I might have actually checked it out on my own beforehand, but the audience might not have. And I wanted to ask you more specifically about the relationship between how you think about the relationship between your own research and gaming, and then further pose the possibility of gaming as itself a form of scientific work.
Practice, So, well, I don't know if the audience is familiar with scientific games, but there was a very famous game called Fold It, that basically was like
that as well.
It was like a puzzle game where you have a protein 3D structure, and then you use your intuition to basically find the 3D structure of the protein. And everyone got excited in biology, like as a researcher, and everyone was like, wow, this is amazing, because people can find and discover things and then publish, etc. And everyone was saying this is democratization of science. I didn't think it was democratization of science, because it's just using people to perform a task.
It's free labor.
And people are not understanding what they're doing. And so what I wanted to do with this game is to actually educate people and offer them a playing ground to play with the idea of genetic engineering, so that they develop intuition for the rationality behind DNA design. And so it was highly inspired by Minecraft, because when you look at Minecraft, people do amazing stuff and they develop communities. And so I said, let's just put like DNA pieces and see what people can come up and they can do something nice.
I want to hear so much more about it, but I think we're being ushered off the stage. Hopefully we'll get to talk more later at the end. All right, thank you. You can't die in Svalbard. You can't be born either. Longyearbyen is a coal mining town whose mines have recently closed. It's the only permanent settlement in a Norwegian archipelago in the Arctic Circle called Svalbard, and is the northernmost town in the world. In Longyearbyen, an influx of scientists has begun replacing local miners, propping up a zombie economy with international research grants. Researchers arrive here in perfect irony to study the Anthropocene. As all the Arctic coal mined since 1901 combusted, atmospheric carbon dioxide levels rose, and the glaciers whose tongues and feet and snouts had waxed and waned along the depressed arêtes of Svalbard for hundreds of thousands of years receded. Their mass balances shifted, they calved and let loose, and the temperatures rose and the waters rose, unleashing a terrible positive feedback cycle. What will happen as these glaciers recede, as the climate warms, and as ecological catastrophe joins geopolitical catastrophe to make this and every other place precarious and unlivable? To find out, I flew to Longyearbyen. Less than 1,000 kilometers, from the North Pole, over 60% of Svalbard's landmass is covered by glaciers. Permafrost renders the ground nearly impenetrable. In the 18th century, Russian trappers would leave their dead above ground throughout the winter, as the earth only softened enough to bury them in spring. And once a body is buried, Arctic temperatures keep it from decomposing. If buried above permafrost, as in the example above me, the thawing ground eventually forces bodies back up until they surface again. So the dead remain fresh, just below the frozen earth, perpetually waiting to be disinterred. I arrive in Svalbard at the end of February, when the sun hasn't risen for some time. In the polar circle, 4 months is spent in total darkness. For another 4 months, the midnight sun never sets. Technically, the sun rose a week ago, but at this time of the year, it never climbs above the mountains on the other side of the fjord. And so, there's a perpetual twilight, a prolonged late morning sunrise that gives way just an hour or two later to early afternoon sunset, staining the mountaintops pink. It's not overcast, really. The light is limpid and the sky is cloudless, but the sunless half-light makes every hour seem untimely. Now, the untimeliness of life is at the heart of my story, and while I will soon focus on life frozen, either naturally in the Arctic Circle or via technical means, I'd like to pause now to direct your attention to a different sort of resurrection. Indeed, I will now resurrect life, or attempt to. The Rose of Jericho is a plant native to the deserts in the Middle East, places like the Negev Desert, but also parts of Syria. And when desiccated, it can last for decades or even hundreds of years in a state of suspended animation. So for all intents and purposes, this is a plant that is dead. And that suspended animation is triggered by what is called programmed cell death, or apoptosis. But if you add water to the roots of the Rose of Jericho. As the water soaks into its roots, proteins begin to hydrate, and they release substances from a dead seed coat. So as you can see, the plant gradually returns to life. This one having just been watered, think of this as a sort of time lapse. So this is the Rose of Jericho having been resurrected 0 hours ago. This was 2 hours ago, 4 hours ago, 8 hours ago, and 12 hours ago. Now, once the plant has been resurrected, it then releases new seeds into the desert, into what is called the seed sphere. And this plant has long been understood as a miraculous seed, one that is venerated by Christians, Jews, and Muslims alike. There's a reason why it's called the Rose of Jericho. It's also referred to as the resurrection plant. And it's used for many other purposes, from the occult to many early modern and medieval materia medica as a treatment for various diseases and as a way of increasing fertility. But despite this long history of the Rose of Jericho, the reason why I bring it up now is that it is now being sequenced, and its genes are being synthesized by molecular biologists. The reason why they're doing so is because they hope that they can use the proteins and enzymes in the genome of the Rose of Jericho to protect crops against increasingly hostile global climates. The idea being that we could make new crops that will also have the same capacities, that would be able to survive long periods of drought and then resurrect again. But let us return to the Arctic. I forgot that I had a time-lapse video that I was going to show you. Well, you can go on YouTube, just Google Rose of Jericho, and you can watch it too. Okay. So let us return to the Arctic. The cemetery is surprisingly hard to find. 3 times, I mistake fields of wooden stumps for the cemetery. These narrow poles cant acutely from the ground, a kind of German expressionist gesture toward a cemetery. I later learned that these are the piles on which the original homes of Longyear City, what is now known as Longyearbyen, were built in 1906. German forces shelled and burned them during World War II, but they're still here. And the reason why they're still here is because since it doesn't thaw, water can't rot the wood. I nearly miss the actual cemetery, though, unable to see it until I'm already there. Rows of white wooden crosses barely visible against the snow-covered mountainside. No one has been buried here lately. The highest row of gravestones mark the graves of 7 miners who died of the so-called Spanish Lady, as the 1918 flu was then known. The very young and the very old are usually hit hardest by the flu. Yet World War I cultivated one of the deadliest viral strains in history. As young men marched across Europe, the so-called Spanish flu preferentially culled young adults. The results were devastating, a horror show of pandemic epidemiology clutching at the coattails of military maneuvers and socio-political ruin. On September 24th, 1918, 69 young men, most of them fishermen or farmers, traveled on board the Forset from the Norwegian mainland to work Spitsbergen's coal mines. In the 3 days it took for them to sail from Tromsø to Longyearbyen, every last one of the passengers fell ill. 7 men died within weeks of their arrival on the archipelago, and the oldest of these was just 28 years old. Their deaths are recorded in the diaries kept by Stora Norska, which is the local coal company that had purchased Svalbard mines from the American-owned Arctic Coal Company 2 years prior. Stora Norska buried them here, on this stark and desolate hill overlooking a town that they would never see. Presumably to save money, their bodies were wrapped like fish in yesterday's newspapers. The bodies of the 7 miners remained frozen beneath Plateauberget Mountain for 80 years, until forensic pathologists and archaeologists, armed with new DNA sampling technologies, arrived in Longyearbyen to find out whether the flu virus that had felled them was still biding its time in the Arctic tundra. Researchers had already attempted to exhume victims of the 1918 flu in other parts of the Arctic and in Iceland. So as early as in 1951, microbiologists had traveled to Brevig Mission, which is near Nome, Alaska, to exhume the bodies from a mass grave of native Alaskan flu victims, though they were unsuccessful in those efforts. 50 years later, after exhuming 6 of the 7 bodies from Longyearbyen Cemetery, Canadian medical archaeologists sampled soft tissue using a boring device that had first been developed in forestry for taking tree core samples. So they used this boring device and inserted it directly into the corpses of the people who had arrived to mine the coal mines. This human tissue, of course, would have long since putrefied in bodies that had been buried in warmer ambient conditions. But in Longyearbyen Cemetery, the young men's corpses were desiccated and frozen solid. 100 samples of lung, liver, kidney, and brain were dissected from the bodies. And the genetic material of the 1918 flu virus, researchers found, was still there. Bits of ribonucleic acid, or RNA, fragmented and burrowed inside the bodies of these Norwegian fishermen. Further, the bacteria clinging to their lung tissue was also still alive, and researchers were able to cultivate it in the laboratory. It grew heartily in nutrient broth when it was heated to body temperature. 7 years after the bodies were exhumed in Svalbard, the genetic sequence of the so-called extinct virus was revived, not from the Svalbard samples, but from a different piece of human tissue that was kept frozen in a pathology laboratory in Washington, D.C. The sequence was then confirmed using the corpse of an indigenous Alaskan woman, again, one of the people who had been preserved in permafrost near Brevig Mission, Alaska, in a mass grave. So by that year, the entirety of the sequence of the 1918 flu was published in Nature and online. And researchers at the CDC in Atlanta who resurrected the virus then sprayed it into the noses of mice. All of the mice died within days. The Spanish lady's resurrection ignited a furor among biologists and ethicists who were horrified by how easy it was to revive this latent flu. They worried that a bioterrorist could easily download the sequence from a public database, and it was open to the public, that they could order the virus from a DNA synthesis company. As we learned, DNA is very cheap to buy online, and then they could have it shipped back to them via FedEx. But what does it mean that newspapers and science journals report that we are now haunted, to use their language, by the specter of a virus that once lived, died, and was brought to life again? Attending to living things that can be arrested and revived reorients definitions of life away from the continuous unspooling of generations and living phenomena. So freezing does not simply pause, reverse, or restart biological time. Rather, freezing grafts the past onto the present in order to capacitate future life forms. So I'm now going to resurrect my voice. One moment, please. My second story attends to a different sort of condensed vital potential awaiting resurrection, literal seeds. In 2003, the World Bank allocated $13.6 million to update 11 gene banks, which collectively held over 600,000 agricultural genetic resources. Mostly when I say genetic resources, I'm referring to land races. In 2005, just when the gene banks had been finally updated, Hurricane Katrina hit. And researchers began to wonder whether agricultural diversity could ever be secure in cities that were vulnerable to ecological disaster. They realized that gene banks were located in places that could be quickly dismantled by different kinds of geopolitical strife and natural disaster. Such gene banks included Nigeria, Colombia, and Nepal. Now, years earlier, Norwegian scientists suggested burying a gene bank here in a Longyearbyen coal mine. A vault dug into the permafrost in Svalbard suddenly seemed as safe a place as any, and perhaps safer than most. A thin cement wedge piercing the frozen mountainside at a steep incline, the vault's brutalist exterior suggests how deeply it is rooted beneath the earth. Above the doors and along the roof is an installation of prisms and fiber optic cables that reflect the midnight sun in the summer and glitter like the aurora borealis in the middle of the polar night. It looks like a science-fictional post-apocalyptic bunker, which is, in fact, exactly what it is. At first, the Doomsday Vault seems to keep a kind of evangelical time. What I mean by that is millennial anticipation oscillates between apocalypse and salvation, with nothing in between. Millenarians either welcome Doomsday with open arms, or they stockpile beef jerky and Bitcoin, and sometimes they do both. The point is that the end is nigh, and the present has been reduced to a horizon in which to anticipate the end of days. This notion sounds somewhat like Walter Benjamin's point about perpetual catastrophe supplanting dialectical history. But more importantly, it is also the pervasive attitude of our times. Just turn on the news. The vault's engineers have prepared for a day when the permafrost has thawed. They've accounted for a 70-meter sea level rise, which is a rough estimate of what would happen if every single glacier in the world were to melt. They compounded that scenario with a tsunami, and then they built the vault 5 stories above that predicted waterline. Engineers calculate that given the current rate of climate change, the vault would remain below freezing, that is naturally below freezing, even if the electricity were out for the next 2 centuries. How long did you build it to last? I asked my guide. Essentially forever. So allow me to escort you deeper into the vault. The door to vault number 2 is overgrown with frost, which has crystallized around the door frame and bloomed across the door handle. My guide opens the door, and minus 18 degrees Celsius is a sucker punch. Yet here is abundant life, 860,000 different varieties of crops, and 120,000 different strains of rice alone. Seeds are sealed in triple-ply, puncture-resistant vacuum packaging and are then loaded into plastic crates, which are stacked on shelves. Looking inside one box, I find ampules of squash and bags of anise. Every major crop in the world is in this room. Not just wheat, oats, barley, potatoes, lentils, soybeans, and alfalfa, but also heirloom seeds and forgotten land races. Boxfuls of foraged grasses are stored cheek by jowl alongside sorghum, foxtail millet, burr clover, purple bush beans, pigeon peas, Kentucky bluegrass, and creeping beggarweed. Every country in the world is represented, as are several countries that no longer exist. Colombia, North Korea, Russia, Taiwan, Ukraine, Switzerland, Nigeria, Germany, Israel, Syria, Zimbabwe, Tajikistan, and Armenia share shelf space in this pastoral League of Nations. On February 26, 2008, the day the seed vault opened, Pakistan and Kenya were first in line to store their seeds. The previous year, the disputed election of Mwai Kibaki in Kenya triggered ethnic violence against the Kikuyus. Karachi had catastrophically flooded and was also seen to a bloody suicide bombing, and Benazir Bhutto was assassinated in the streets of Rawalpindi. Perhaps, for Kenya and Pakistan, a cache in the seed vault is a way to refuse political and climatological vulnerability, or even to forecast a future that might sustain life. I notice that one shelf of the vault is half empty. 4 years into the civil war and humanitarian crisis in Syria, violence barreling northwards toward Aleppo jeopardized the headquarters for the International Center for Agricultural Research in the Dry Areas, which is thankfully has an acronym, or it's called ICARDA. Hundreds of thousands of seeds were banked in ICARDA, including some of the earliest strains of Levantine wheat and durum, many of them more than 10,000 years old. Now, in an Aleppo summer, a power outage would thaw these seeds much faster than would one in Svalbard, so ICARDA shipped the seeds to Svalbard for safekeeping. The Syrian gene bank, now relocated to Morocco and Lebanon, recently requested 30,000 samples from its original collection to help rebuild the country's stores of barley, lentils, and chickpeas. Like the viruses that are thick on and under the ground in Svalbard, the vault condenses multiple temporalities. These are places in which some subcutaneous latent life, a virus, a seed, erupts into the present or is buried for future use. Coldness synthesizes and reassembles biological time. Following theorist Dipesh Chakrabarty, latent life in Svalbard is a sort of time knot that refers us to the plurality that inheres in the now, the lack of totality, the constant fragmentariness that constitutes one's present. The 1918 flu is a specter of the past that has erupted into our present. The seeds in the Svalbard Global Seed Vault are artifacts of our present that are now buried for future disinterral. I suggest that the temporalities in which things in the Arctic Circle move or don't move, and the speed at which they do or do not do so, is itself a principle by which things can transition from life to immortality. Some life is slower than molasses on a snow day. Some life is stuck in the past, and much of that old life is durable in its endurance, perpetually present in the present. It lags. It persists. Latent life promises to last forever, even as it is perpetually endangered. It can be patient as a saint. It can be quiet to the point of cryptic. This fact points us to the possibility that rather than being the common denominator underlying all living things, life is perpetually a problem of temporal discontinuities. Viruses and seeds that are stilled in states of suspended animation are classed as only problematically alive, but not quite dead yet. Biding their time, awaiting resurrection, they are pervaded nonetheless by a potent and vital potentiality.